Welcome, everybody. Welcome to Chestnut Chat. Again, I am Sarah Fitzsimmons. I'm the Chief Conservation Officer with the American Chestnut Foundation. I am joined today by a special group of folks. This is, as Terry tells me, this is the inaugural presentation of over 40 years of work on roundleaf birch. Is that right, Terry?
50 years of work.
50 years of work. So this is the first time this information has ever been seen, and you're going to see it right here on Chestnut Chat. A few housekeeping notes before we get started. First, if you're not one of the 6 panelists that you can see here, we can't hear you, we can't see you. Have lunch, answer the door, watch Price is Right, whatever you need to do. We can't see you. Even if you try, it ain't happening. So you're all good. If you have some witty banter, some chat, let us know where you're at. Let us know what the temps are. Let us know what's going on outside. I'm really curious. If you signed on late, you didn't see who's Griffin is in the Grand Caymans at 78 degrees. I think that's the current winning high temperature. 17. I haven't kept track of the low temperature, but I think the high temperatures are of a little more interest to me, at least today. So throw that in the chat. If you have a question, throw that in the Q&A. I see a question has already popped up, although that's John just testing. Thanks, John. What we're going to do today is what we often do, which is Terry is going to go through his presentation. It's going to take about an hour. So I invite you all to sit tight and listen to him. Wendy, I see your hand raised. I typically don't call on folks, but if you want to throw something in the chat or put something in the Q&A, please just let us know. And then once Terry's done, I will go through the Q&A. If you throw a question in the chat, it's likely to get lost. So please keep your questions in the Q&A, witty banter in the chat. I will hold all questions pretty much until the end. Unless there's if someone's super confused, throw that caps. I am confused, interrupt Terry and ask him this question. Otherwise, I'm going to hold it until the end. So I think thanks again for joining us today. Next month we are going to be talking about chestnut weevils. Is that right, Vasily? You helped set this one up together.
Yeah, that's right. Next time is March, so I believe that's going to be Camilla and Jonathan. Yeah.
Yeah.
And
So
Asheville.
they're from UNC Asheville. That's going to be March. So we're going to do something a little different in March. TACF has their spring in-person meetings in Abingdon. So we're not going to have it on the 3rd Friday, which is tradition. We're going to have it on the 4th Friday. So mark your calendars for March 28th. You will get several e-sprouts about this, but March 28th, we're going to talk entomology and American chestnut. So that should be a really good session. Camilla and Jonathan also recently got some finished out a round of funding from TACF's external grants funding. So we helped fund this work, and excited to hear what they found through that research, and that is ongoing. So join us next month for March, as you do always. This presentation is being recorded, so if you say anything, panelists, it'll be recorded. We'll everything up until I say, hi, I'm Sarah Fitzsimmons, that'll be cut out, that'll be edited. It'll be put up on our website, tacf.org/chestnutchat. It'll take us 3, 4, 5 days to put everything up. Hal's probably here. Hal does all of that work. Thank you for putting that together. It'll be up on our Chestnut Chat website. It'll be up on the YouTube just a couple of days from now. So if you missed anything, it is recorded, and you'll be able to have access to that very shortly after the session. So I want to thank you all for joining us. Terry, I'm going to hand it over to you to make an introduction of yourself, and then the other panelists as we move along. I really appreciate it. With that, take it away, Terry.
Well, good morning from the banks of the Huron River in southeast Michigan, land of the Ottawa, Ojibwe, Potawatomi, and Wyandot First Nations. His land was ceded to the U.S. government in the Treaty of Detroit in 1807. Right now, it's pretty iced over. If you can see my image, that's in fact the Huron River behind me. We have 50 years is a lot of time, a half century, and so there's a lot of material here to go through. I'm going to, this is as Sarah indicated, a lot of this will have been seen for the first time. When I put the thing together, I put the story together, not being concerned about the length of it. And so it is what it is, but it is longer than probably it should be. But hang on to your seats. It's designed in such a way and set up in such a way that it's fully explicable, hopefully, just by looking at the slides, even without my voicing. And so the information should be there. The downside, of course, is that it appears to be pretty dense, as you'll see. Now, what I'm going to do is I'm going to first go through and do a section on milestones, starting from the original discovery of the Virginia round leaf birch, Betula uber, in 1914 and go through to the present. And then I'm going to go through the 6 action areas in the recovery plan that are designed, when fully implemented, to bring about the delisting of Betula uber. And then I want to wrap up with some next steps and conclusions. So that's where we're headed. So hang on. A lot of material here, and hopefully we'll have enough time for questions at the end. But also, obviously, I'll be able to see your responses too in the that you post, and then I'll have my contact information at the end where you could contact me and we could carry on some more conversations. One of the objectives in all of this is to get more people involved in this effort, and there are opportunities for that. So in terms of acknowledgements, and these are just people who have been involved since 2021, since we re-entered with our activity. And so Tom Blevins with the Forest Service was for many years watched over the progeny or additional populations. Bob Ford from Frederick Community College is now retired, was a graduate student who did work on dispersal of brownleaf birch with me when he was a graduate student at Virginia Tech, and has continued to do most of the field work with me currently. He lives in Asheville, North Carolina. Amaryllis Rosari and Emily Argo are endangered species specialists with the Virginia office of the U.S. Fish and Wildlife Service in Gloucester, Virginia, and they have responsibility for the managing the Virginia roundleaf birch. Richard Cray and Cal Peer at the Redall Homestead Research Center in Kreitz, Virginia, was the place where the seedlings that were used for the progeny populations were produced and brought to the site. Vasily Lukoba, who many of you know with the American Chestnut Foundation, has been great in terms of his providing staff to help with some of our field work, and in particular our catkin counts. Doug Ogle, retired from Virginia Highlands Community College, was the one who rediscovered Virginia roundleaf birch in 1975. Chris Shaw and Brittany Phillips with the Forest Service, Mount Rogers National Recreation Area, where the population resides. Chris has really done a lot of the field work and follow-up field work, and Brittany on the NEPA process approval. Kim Shear with the Morton Arboretum did dispersal work, among other things. More recently, seed dispersal work, I'll show you. Justin Showalter and Butch Shaw from RX Fire Effects did the treatments, the regeneration treatments. It's a tremendous amount of work, and we really appreciate that. Suresh Subidi at Norfolk State University, as among other things, an ecologist, but GIS specialist, and provided that expertise. Keith Tigner with VDACS, the Virginia Department of Agriculture and Consumer Services, oversees the Virginia Endangered Plant and Insect Act. And unfortunately, he's not able to join us today, but they're a really important partner in this. And finally, Hiram Way from Michigan Tech is a forest geneticist with a specialization in bioinformatics, and we've been in conversation and move forward in that area. So with that, I'm going to just turn it over for a minute to Ami Arizari, and then in turn, Emily Argo, to say just a few words about their perspective on the Virginia roundleaf birch. So Ami.
Yeah, thank you, Terry. Like Terry said, good morning. My name is Ami Irizarry. I'm a biologist with the U.S. Fish and Wildlife Service. And my involvement with the Virginia roundleaf birch, I used to be the recovery lead species for the birch in the Virginia field office. And in 2020, I wrote the 5-year status review for the species, which led me to connect with Dr. Terry Sharik in 2021. And we started working on recovery efforts for the birch back then. We gathered with a group of scientists, natural resource management managers, and other interested parties to discuss the status of the species and develop a conservation strategy to pick up recovery efforts for the roundleaf birch that, like Terry's going to explain today, had not had been many at that point. So we were able to secure some funding and do work to implement management actions to promote natural regeneration and monitor for the species, which at that point hadn't really occurred in over a decade. So I now have a different role with the U.S. Fish and Wildlife Service, and I'm not able to continue leading the recovery efforts for this species. So I'm really happy to pass that role on to my dear colleague and friend, Emily Argo, who I'm sure she will continue to do a great job working towards recovering and delisting the species. So Emily, if you'd like to say a few words.
Absolutely. Thanks, Ami. Good morning, everyone. I'm Emily Argo. I'm at the Virginia Field Office in Gloucester, Virginia. And I am stepping in to fill Ami's shoes here as the recovery lead for this species. And so we're trying to take the next steps with continuing the great work that Ami had funded to start working towards some of the recovery goals and tackling some of the recovery actions identified in the recovery plan to keep momentum going with this species. So I'm hopeful that we'll be able to keep some more projects and some more research going in the future. Thank you.
Great, thanks to both of you. This is this was posted before in background reading. The Virginia Forest asked me to write something on concerning recovery efforts with the Virginia roundleaf birch, which I did, and you have access. That's available online, and it has a part of what I'm going to talk about today, but not all of it. But some of it is in more detail than what I'm going to talk about. So I still refer you to that. So let's look at milestones from 1914 on to the present. So in 1914, on June 14th in specific, the Virginia roundleaf birch was discovered, first described and discovered by William Willard Ashe along the banks of Dickey Creek in Smith County, Virginia, at about 2,800 feet in elevation. And he considered it to be a roundleaf variant of the common sweet birch or black birch Betula lenta, and thus named it Betula lenta variety Uber Ash. And then in 1945, Merritt Fernald, who was the director of the Gray Herbarium at Harvard, examined Ash's specimens, and he made the decision to elevate Virginia roundleaf birch to the level of species, thus Betula Uber Ash Fernald, and transferred it from the dark bark tree birch section of the genus to the shrub section, presumably because roundleaf birch, shown here, and Betula lenta above it, has leaves which are similar to the shrub birches. So he made that decision to make that change. And I just have a note here at the bottom then, over this period of time, between when Ashe published his first description in 1918 and through 1982, the species has been this taxon has been variously classified at the species level, the subspecies level, the variety level, and the form, all the way down to a local variant of form over that period of time. And I think you'll understand why when we proceed with that. In the Smithsonian Institute in 1974 and the U.S. Department of Interior in 75 rendered the species probably extinct. And so people just thought it was extinct, in fact. And it wasn't until 1975 that Doug Ogle, who again was at the Virginia Highlands Community College in Abington, rediscovered the Virginia roundleaf birch along the banks of Cressy Creek I've got to move my, excuse me, I'm just getting blocking from the names here on my screen. Banks of Cressy Creek at 2753 feet in elevation, so very similar. And that watershed, Cressy Creek, is immediately adjacent to, immediately to the east of Dickey Creek. And what we've concluded, and we've gone on to do enough searching to look that in fact, that probably was the case, that Ash was in error and thinking that he was on Dickey Creek, when in fact he was on Cressy Creek, just to the east of it. So in any case, it consisted of a population when he first saw it described of 34 individuals, and you see 12 adults, a sapling, and 21 seedlings, and the adults range from 25 to 46 feet in height and 1.5 to 5 inches in diameter breast height. So, and I put here at the bottom, then 1975, I joined the faculty in the School of Forestry and Wood Sciences and Wildlife Sciences at Virginia Tech. I had received my PhD at the University of Michigan in 1970, having worked on the ecological genetics of dark bark birches, tree birches in the southern Appalachians. So as luck would have it, I was there and predisposed to getting involved in this stuff. And so further in 1976, after some more reconnaissance, we were able to, we determined that there were actually 41 individuals in the population, and that included 18 reproductively mature adults. Most of all, and this is really important, we're already in the subcanopy, and 23 subadults, actually seedling sapling size age, and they were all under a closed canopy. The adults range from age 3 to 45 years at that time, and they were distributed over a 700-meter Sugar Grove, Virginia is up here in the northwest corner, distributed from this point here downstream, upstream for 700 meters, or about a 4/10 of a mile along the banks of Cressy Creek, shown at the bottom there, in a band that was really a highly disturbed second growth forest, about 60 meters or 200 feet wide, surrounded by residential and agricultural land. And you see that in this image right here. And so this is a highly disturbed area, and it's bounded by residential and agricultural land, so not a lot of room to move. Most of the trees that you're looking at in this photo along the bank are Betula lentas, or sweet birches. And I would also point out that these different colored dots here show 3 contiguous ownerships. The first up here being the Forest Service ownership, and that with the green dots here, this is in 1977, by the way, and then the first private ownership here in purple dots, that includes 1 adult individual and the rest seedlings. And then the second in yellow, the second private ownership, which included the adults at that time. In 1977, we formed the Betula Uber Protection Management Research Coordinating Committee, and it's had up to 23 of 45 members at any one time, in total at any one time over the next 2 decades, from 10 organizations, including public and private sectors, with the U.S. Forest Service taking the lead on this. And in 1978, then, the Virginia roundleaf birch was listed as endangered under the Endangered Species Act of 1973. It was the first tree species so recognized. Interestingly, the to take clause did not apply to plants until 1982, and then only on federal lands. So it was almost a decade before the to take clause, but only on private lands. And this all goes, really interesting phenomenon, goes back to British common law, or the so-called King's Law, where if organisms have the ability to move, they belong to the state, to the king, or translate the state. If they don't move, then they belong to the landowner. And that has carried through to the present, where we have very different situations with the protection of animals and plants, because the state owns, in essence, owns the animals, they don't own the plants. because of the the listing, then the U.S. Fish and Wildlife Service joined the committee, and that's when really, even without earmarked funding at that time, conservation efforts began, and we fencing, stream bank stabilization, search for additional populations, asexual and sexual propagation, all of that without earmarked monies. In 79, it was listed. I'm sorry, we just did that. It was listed as, it was listed as endangered under the Virginia Endangered Plant and Insect Act of 1979. And here the to take clause is a little bit stronger. It applies to both public and private lands, except for private landowners. So if you've got a endangered plant species on your land, you can do anything you want to it. But that caused then brought about the BEAT Act, the Virginia Department of Agriculture and Consumer Services, to join the committee. And those three entities have been really important: the Fish and Wildlife Service and the U.S. Forest Service and BEAT Acts, major players throughout this period of time. This led to the first earmarked funding by the Fish and Wildlife Service of $10K to develop a recovery plan. And the first it's been about, I count, about $270K. It's been earmarked over the years. So not a huge amount of money by today's standards, certainly, over that period of time. And at that time, then research really accelerated to include monitoring of flower and fruit production, seed collection, seed dispersal, greenhouse and field seed germination, morphological analysis of leaves and fruits, wood anatomy analysis, controlled crosses involving sweet birch and roundleaf birch, and vegetative analysis of the roundleaf birch population site. And we also, at that time, introduced an interpreter program at the site of the natural population and at the U.S. Fields Office, Forest Service Field Office nearby. In 1982, the Fish and Wildlife Service approved the recovery plan. It was then revised in 86 and 90, followed by 5-year reviews intermittently through 2020. It was the first recovery plan for an endangered species under the Endangered Species Act of 1973. So it took nearly a decade then for the first recovery plan of a plant under the Endangered Species Act. And overall, the goal was to increase the number of roundleaf birches in the wild to a point where the species could be delisted. That's what we're interested in here, is delisting. And it was estimated, we initially estimated 1,000 individuals in each of 10 populations, but we later revised that to 500 to 1,000 individuals greater than 2 meters in height. And so at the core of this recovery plan, there are these 6 actions put forth to accomplish this goal to reach delisting. Maintenance expansion of the natural population given the highest priority, establishment of self-sustaining additional populations in a wild, a priority 2 item, along with continued searches for other populations, and then determination of systematic relationships with other birch species, along with retention of germplasm through cultivation as priority 3 items, and then finally implementation of educational programs, priority 4. 1986 through 88, I moved to Michigan Tech, 1,000 miles away from the population, but remain involved. And during that period of time, we developed the Virginia Roundleaf Birch Data Management System, and it became operative in 1988. It included all the data collected on the natural population and additional populations since 1978. Now, here's an interesting thing. I don't know if any of the others have experienced this, but nobody told me that these floppy disks would degrade. I now understand that after about 8 years, they degrade. And so that data is not currently accessible due to degradation of those floppy disks. We're trying to still go about recovery of the information. I think we have most of the original data, but would all have to be re-entered again. It will take some effort and time to do that. I then, in 93, I relocated to Utah in December, where I resided through 2012, and that was 1,900 miles away. And I also took on an administrative position. So that really made it very difficult for me to continue. And another really important factor here, Pete Farrett, the forest geneticist at Virginia Tech, my colleague who was working with me, passed away unexpectedly in February at the age of 46. And this is not about us, not about me, but it's just an example of how people make a difference. They really do. And when Pete passed away, it really had a major impact on our efforts related to the genetic component and propagation efforts, as you'll see later. 1994, because of our efforts of establishing the additional populations, as they were called then in 1984 and 1987, it was downlisted at the federal level from endangered to threatened. And then at that point then in 1984, the earmark funding largely dried up. With limited amounts through 2003 for monitoring and maintaining the additional population. And part of that drop off was influenced by our research finding that Virginia roundleaf birch is likely not deserving a species rank, despite the Federal and State Endangered Species Act allowing for protection below the level of species. And you may allow the snail darter, you may have read recently where the snail darter, people are saying, well, we put all this money into the snail darter recovery, and it turns out it's not a quote unquote good species. Even then, of course, this is still under the act, it can be considered. We can come back to that. 2012 to 2024, I returned to Michigan Tech in 2012, 1,000 miles away, but then I retired in 2018 from Michigan Tech and relocated to Southeast Michigan. So now I'm about 500, a little bit over 500 miles from there. I am retired, by the way. I'm not an employee. I'm a emeritus professor, but they also allow me to have the title of research professor, which allows me some access to funding opportunities I may not otherwise have, and also makes me seem like I'm not as far over the hill as people who are emeritus faculty, right? So that's another factor. The first in 2023, I came back, the COVID hit. I wanted to get re-involved in 2021. I did in 2021, and we then started measurements of mortality, growth rates, and catkin production. And then that led to the first implementation of regeneration treatments in what we were called then the additional populations. There's a language being used by the Fish and Wildlife Service, and now we mostly refer to them as progeny populations, with some funding from the Fish and Wildlife Service and VDACs. So we had 4 decades after the approval of the recovery plan, and between then and the establishment of populations. And indeed, in 2024, as you'll see, we've realized our first natural regeneration of Virginia roundleaf birch in these progeny populations. But with the original population now down to 3 individuals, by the way, which you'll see. So let's turn to maintenance and expansion of the, let's see here. I'm getting a little bit of a lag here. Maintenance and expansion of the natural population. And I think we need to start with a little bit on the natural history of dark bark birches in North America. This genus worldwide consists of 30 to 60 species, depending on who's doing the classification. But in North America, we're focused on this subgenus of dark bark tree birches that is distinguished by having high concentrations of oil wintergreen in the inner bark and erect female catkins at maturity, which I'll show you in a minute here. They're short-lived early successional species in temperate regions. Most of them, they may, in close canopy situations, reach a couple hundred years of age. They've been known to go up over 300 years of age, but usually around a couple hundred years. This group is confined to the eastern U.S., the Appalachians, and the Lake States, consisting of 3/4 species. River birch, Betula, and niger used to be included in group, but it hasn't. So the only quote-unquote 3 species are Betula lenta, sweet birch, Betula allegheniensis, yellow birch, and Betula uber, Virginia roundleaf birch, all of which co-occur or sympatric. They typically are found in cool, moist, montane conditions in coves and bordering small streams in the southern Appalachians where the native population is from, but also interesting on rocky outcrops. And then as you move north, they move out more, they're more widespread on the north. They require, as a really important component, disturbance to regenerate, and i.e. a positive overhead cover and leaf litter. And they're moderately intolerant of closed canopy, closed forest conditions. Another really important factor in this. So in terms of their reproductive biology, the birches are monoecious. They have male and female flowers on the same individual, and those flowers are inflorescences that are called catkins. It's a single central axis that bears sessile or non-stalked flowers of one sex along that axis. And so here in the upper right, you're looking at a mature male catkin that is pendant, is hanging down in spring with leaf out. If you saw that in the winter, you would be looking at this in the lower left. There are these small pencil-like things which overwinter there. They're actually differentiated the previous season, and then when the leaves fall off, you see them. They're very characteristic, in fact, and allow us to do the counting. The females, on the other hand, are contained in what appear to be normal vegetative buds, and they don't emerge until the leaves begin to flush out in the spring. So you get the males producing, again, this is all wind pollinated. So these male flowers are just really a cluster of stamens. And then the female flowers are over here, and you'll see three of them associated with this so-called bract. And the interpretation is that this once was and this depicts various genera. There are 6 genera in the Betulaceae, the birch family, 5 of which occur in North America, and they're distinguished on the basis of what happens to the evolution of this primordial inflorescence that got reduced in various ways. And in the case of Betula, you have these 3 flowers on a side axis here with 2 leaves subtending them, or bracts, as they're called, leafy bracts. Most of those get wiped out except for 3. They get reduced to the bottom area, and they end up with this where we're looking at here below, where these 3 bracts coalesce. These 3 bracts coalesce into one 3-lobed bract, and then subtended at the base of them. Then there are the 3 flowers, the 3 female flowers, which then mature into winged nutlets, or they're called, or chimeras. And each one of those has a single ovule, usually a single ovule within it. So, you know, the male flowers the males are flowering in the spring before leaf out, and the females are mature and dispersing in the fall after leaves. We don't want leaves in the way, obviously, when you're when wind is your mechanism of dispersal. So the wind dispersed pollen again in the spring, and wind dispersed chimeras in the fall and in the winter. Actually, most of the dispersal in our area occurs between about mid-December and mid-February, and extends a little bit beyond that. But here we have a single female catkin that's mature in the fall, and these naturally disintegrate. So this thing falls apart. Here's the remaining axis here. And here are the bracts, these 3-lobe bracts that I talked about. And then here are the winged nutlets, or chimeras. And these things are less than 3 millimeters in both width and length. So they're really, really small. And there are, on average, there are about 100 bracts in each catkin, and therefore about 300 chimeras. So, you know, a given tree may have more than 1,000 catkins on it, female catkins. So we're talking about 330,000 seeds from one individual. So the seed germination, seed emergence occurs in early spring under cool, moist conditions. and then rapid growth in the near absence of overhead cover following that, assuming the substrate conditions that are conducive to that happening are there. That is, most of the overhead canopy gone and leaf litter gone. So in essence, mimic what you would expect from fire, or in some cases logging will do the same thing, but it's not as completely. So so here the population here is a you saw the image earlier here. Here's that same population with the Forest Service land here. 370, tree 370, which is shown here on the right, is the largest individual and the champion, quote unquote, champion tree in that population. And this is Forest Service land here with there's in 1977, two individuals. And then down here, a cluster of individuals on the first private land ownership, and then another cluster on the second plant ownership. So we went in 1976 from 41 individuals to a year later to 26 individuals. That loss was mostly subadults due to human factors, including removal of individuals for study and propagation, transplanting by the private landowner. Again, the landowner can do whatever they want, and vandalism. And so we went then to that number of individuals that you see here. And most of that loss early on was in this juvenile part of the population. And then by 1994, we're down to 10 individuals. And most of that was the loss of reproductively mature individuals in this population here. And due to natural factors, in contrast to the earlier stuff, these things were already in a subcanopy position and destined not to live for a long period of time. And then by 1921, we were down to 3 individuals with no documentation of that loss because it wasn't being monitored over that period of time. And so we now have only 3 individuals that remain in the natural population. In addition to these 2, a small understory individual that emerged some point later in time is near here, and we're trying to release around that individual to give it some exposure to the canopy. If these things do not have direct sunlight, some percentage of direct sunlight, and they move to the understory, they're destined not to live for a very long period of time. We erected protective fencing around both on the public and private land, but that didn't stop the vandalism and didn't stop the vandalism on the private lands. They didn't have these high fences like was on the Forest Service land. This is the big tree 370 right there behind it, beside Cressy Creek. And but it's also worth noting that this area has a lot of birches in it. And so pre-1979 growing styles and season, our inventory showed that there were about 23 Virginia roundleaf birches, 623 sweet birches, and 11 yellow birches, Betula alleganiensis, in that population over that 700-meter span along Cressy Creek. In November, December of 1981, given that situation, we cleared away the woody vegetation and the leaf litter in areas within 60 meters of the potential seed sources. We had done we had studied how far these seeds are dispersed and when. So we used this within 60 meters. And that resulted in 80 Virginia roundleaf birch seedlings being recruited to the population in 1982 growing season on private land, but not on forest land, and probably not on forest land because the distance between the trees to roundleaf birches was too great to affect pollination. I could have mentioned earlier that birches are obligate outcrossers to cut down on inbreeding, strategy to cut down on inbreeding, and you may get 1% of selfing, but it's really low. So if you don't have 2 roundleaf birches within pollen dispersal distance of each other, you're not going to get roundleaf birches. These treatments were done to mimic disturbance such as fire for early successional species. And just a note here that the percent Virginia roundleaf birches, 8%, was similar to the percent of Virginia roundleaf birches in 1976. In the population. So the seedling population was mimicking the adult population in terms of percentages. The number was reduced, this 80 seedlings reduced in a fall to 80 30 seedlings by 83, which was mostly seemed to be a natural sort of thing, but then to zero in May of 1985, all due to vandalism. So we knew that we were in deep trouble here. And so 1982, well, we did selective in this population, selective release around these birches performed by the performed on Forest Service land and on one of the private ownerships to try to create a situation where these individuals would live for a longer period of time. But the other private landowner denied access. That's a whole nother story about a relationship that did not go well between the private landowner and the National Arboretum at Washington, D.C. It's another story. And I'm not being negative towards the National Arboretum. It was a misunderstanding sort of thing. 1984, the Nature Conservancy purchased 14 hectares of pasture land adjacent to the natural population. In turn, that was purchased by the Forest Service, and that became the future site for progeny population 21 that we established. So these failures then collectively heightened the need to establish additional populations in the wild and to mass propagate seedlings for distribution to the public and private sectors. It really got our attention. You can think about this as a form of assisted colonization or assisted migration where human landscape barriers exist. And so that was our next move in this equation. So in early 1980s, we identified 20 wooded areas, except for one pasture area that I talked about. They were selected for clearing of all trees and shrub cover in 20 by 50 meter plots. And they're shown here on the right, those locations. All of them were located within the Crescent Creek watershed, which is about 10 square miles, and all of them on Forest Service land. They covered a wide range of sites where sweet birch is known to occur. Priority given to along streams and in coves, but out onto open slopes, opposing slopes in some cases. And I just had a note here that several of these, in fact, were formerly occupied by American chestnut. And so here we're looking at one of these additional population of progeny populations. We took seed from open pollinated mother trees, 6 roundleaf birch and 4 sweet birch in the natural population. They were germinated in the greenhouse at the Reynolds Research Center in 1982 and 1985. And the seedlings they were held for 2 to 3 years for outplanting in 1984 through 87 at the rate of 5 plots per year. And these plots included, you see them numbered here, 66 individuals on a spacing of 3.3 by 3.2 meters. And we did this to create a closed canopy to try to exclude other individuals, other species from occupying that space. For a total of 1,960 seedlings in all 20 populations. 74% of the transplants were from roundleaf mother trees, and 76% had round leaves. We know the mother trees, incidentally, of all of these. Of course, they're open pollinated, but we know the mother trees of all these individuals. So mortality, and to a lesser extent, we measured growth rates, diameters and heights, and catkin production. That was recorded annually through 1993, and somewhat less frequently to 2003. Most of the mortality in these populations due to natural causes, ice damage, initially deer browsing before we caged them, putting wire cages around individuals when we saw that the deer browsing was really going to be heavy, because when you open these areas up, it's like a magnet, and especially with coppice growth from stumps, sprouts, and so forth, the deer move in and they really begin to do a lot of damage. So, but then later, once they reach sapling size, then you have this deer antler rubbing thing. So we had actually put posts around, steel rods around to prevent that from happening. And then competition for space as well. We had some vandalism. 23 trees across six of those populations over 4 years, from 88 to 91. I'll go stepping forward here. I'll say that once we began to distribute individuals, propagate and distribute them widely, that vandalism went away. And there are no significant, and a key finding here was that there were, in terms of the measurements that we took, there are no significant differences between Virginia roundleaf birch and sweet birch individuals. But and at the same time, we use various methods to maximize survival, including clearing of competing vegetation, fencing around individuals, as I mentioned, and targeted application of herbicides, very little of that. And so here we are in 2023 with one of those populations. The survival among all the populations averaged 56% and ranged from 2 to 89%. That 2% is really an outlier because that was the area, one of our areas that we had originally identified we couldn't get access to later, and so we had to switch off and use that pastured area, the Nature Conservancy property, in an open pasture, and we demonstrated that you cannot establish these things in open conditions, completely open conditions and pasture conditions. So, but 75% of the individuals were in our roundleaf birches, and the other 25% are sweet birches. All mortality since 1921 occurred in suppressed trees in the understory. And so we have the largest individuals now exceeding 11 inches in diameter and 80 feet in height. But there are individuals throughout here that are sprinkled throughout here that maybe are 1 or 2 inches in diameter and maybe 10 feet tall. And they were all the same age, of course. So again, and they're barely hanging on, and then they're just continuing to drop out. The no natural regeneration in the populations, despite at least 3 decades of seed production. We're probably talking about billions of seeds over that period of time. And this is, most likely due to the lack of regeneration niches, this very narrow regeneration niche that these things have. So regeneration of treatments, so then we implemented regeneration treatments in 2023. These areas had to be NEPA approved. I had to get NEPA approval to do that. And we did, and it did in 23, as we'd done previously, counts of male and female catkins and site quality. And that was used to determine which of the populations to treat to secure regeneration, with preference being given to those producing abundant male and female catkins by several individuals on high quality sites. And here you see a tree, and it's probably about 75 feet tall. And if you look real closely, which we have to do, and we use binoculars to count these things, you'll see these male catkins in the top, and here they are. If you could, and then you blow this up a little bit, you see them a little bit more clearly. And then here they are real close. Here's one. They go in clusters at the end of branches from 1 to 4. You see lots of 2s and lots of 3s, these sort of turkey feet. And then, and this, of course, is this is in February, a photo taken in February. And these things will, mature and disperse pollen probably in April, May. And then at the bottom here is a tree with female catkins on it in the fall, just loaded. There are over 1,000 catkins in this tree, dispersed again in the winter. So this preference led to these 5 populations being chosen, but we only had enough funding to treat one, and so we treated one population 3, and it was estimated to produce, having produced over 1 million seeds at that time. And so here's these maps show the abundance of male catkins. So we're out there, in the spring and March, and looking to see what the abundance of male catkins are. And the populations that I just talked about in population 3 have the largest numbers of male catkins. And then, so we use that to go and demarcate the 5 areas, lay out the boundaries of these treatments that we were going to do, not knowing at that time that we didn't have funding to do 5. And then in the fall, then came back and did the counts of the female catkins to make that final decision in terms of the abundance of those catkins. And as a result, then ended up choosing that population 3 that I just showed you. And so what we did was to lay out 6 around this original population of 50 by 20 meters, then with the 96 individuals, 6 20 by meter areas to mimic these sort of large gaps. And here you're actually looking at them with 1 square meter microplot in them that I'll talk about in a minute. But we cleared all of that area of all of the trees and shrubs, and the leaf litter and the substrate was raked to mimic natural disturbances. And here you're looking at, in fact, this treatment area 6 right here is, and we're looking from the back of this treatment in towards the adult population that you're looking at here, right there. And but we had 2 areas, and in particular, this area here, treatment area 3, was less than 20 by 20 meters due to U.S. forest regulation of no disturbance allowed within 15 meters, 15 feet of the stream bank to protect aquatic ecosystems. This is a really interesting example of competing conservation efforts. And interestingly enough, that 15 meters or 15 feet along the stream is the premier regeneration niches and areas for birch populations. And so there's this trade-off between protection of these stream ecosystems versus clearing an area 2 20 by 20 meter strips along the margin of that stream to obtain regeneration. The treatments were implemented immediately after November 15th, which is the first allowable date to take into consideration bat hibernation, but before the start of seed dispersal is starting heavily in mid-December, so you have this fairly narrow window to work with. We then here we see the results of that, and so we sampled these populations in later in September of 2024, and the result of that was over 2.1 birch seedlings per square meter secured, with 15% being positively identified as Virginia roundleaf birch seedlings. And I say positively because when they're real small, it's hard to tell, and we were very conservative. So at a minimum, it was 15% of the individuals, with most of the rest of them being Betulentas. There could be a few yellow birches in there too, which we would subsequently realize. This approximates the percentage of Virginia roundleaf birch trees in the adult population there, and more than twice that in the original population of Betula uber, and equates to about 864 Virginia roundleaf birch seedlings for the 6 treatment areas as a whole, which is more than adequate to support I calculated 270 reproductively mature trees over this area. But looking here, and you'll see here is the number. This is the total birch count in these 6 treatment areas here, and you see the number of individuals, and then these are the number of roundleaf birches, which varied from 1 to 4, and I just indicated they 2.1 birds in per meter squared in this thing on average. So anyhow, and you can see that they're not evenly distributed. So throughout that area, so it really brings up the interesting question: should we perform "quote unquote" assisted colonization or assisted migration on a micro scale and move some of these individuals around? Because clearly they're going to compete with each other given the way that they're clustered. And then if we looked at what that distribution was correlated. These are the results of the seed density based on seed traps in those same 6 areas. And there's some correlation, but it's not strong. And if you, again, if you look at, you go back and look at this treatment area 1, for example, it's low in seeds received dispersed to it, but here it is, and it's quite heavy, in fact, of seedlings in that area. But you do see, of course, that you get this negative exponential movement with the highest seed densities close to the source of the seed sources, and then decreasing away from it. So we then did a bunch of measurements, took a bunch of measurements in the in these 1 square meter plots that we set up that I showed you earlier, and measured live foliage from competing vegetation, leaf litter, soil, rock, water, raw woody debris, decomposed woody debris, stumps, and live stems and rocks, and looked at the correlation with Betula, the density of Betula uber and Betula lenta seedlings shown here. And we had 2 strong statistically significant correlations, and that is a strong negative correlation with leaf litter and a strong positive correlation with rock, exposed rock on the surface. And so in addition to that, we clearing of in 2024, we cleared competing woody vegetation and in mid-November, mostly the coppice sprouts, and we plan to do fencing and transplanting in 2025 growing season. But we do what we could, even if we'd had funding, we could not have done any treatments in 24 due to a lack of catkins. And this was an incredible thing. Here are the males over here in the spring of 24. There was not a single catkin produced on a single tree throughout those 20 populations, which was never done, never had happened in the past. And then if we go to the females, most of them didn't have catkins, but a few did. Now there was no pollen, and so it doesn't make a whole lot of sense to reducing females if you don't have any pollen, right? And so it was a really pretty interesting phenomena. And it's going to be interesting to see how they come out of it this year, and who flowers and fruits more than others, and does it look like past patterns over this watershed. They tend to cluster in terms of their behavior within subunits of this watershed. I don't have time to talk about that in detail, but anyhow, it's an interesting phenomenon. Now, moving to continued searches for other natural populations. Can do this quickly. There was a coordinated search for other populations over a 3-county area in 1977, and that revealed no other populations. So we're really quite sure that the population on Cressy Creek here in this area is the only population that of Virginia roundleaf birch that has existed. Now moving to determination of systematic relationships with other bird species, there have been really a couple really good studies in DNA analysis of the genus Betula as a whole, and in Eastern North American tree species, and there's an indicating that Virginia roundleaf birch is most closely related to sweet birch. I think it's indisputable that that's the case. The phylogeny is pretty clear in that regard. And then cytological studies also show that Virginia roundleaf birch and sweet birch are both diploids. They have 2 sets of chromosomes in their vegetative cells, while yellow birch, Betula alleganiensis, is a hexaploid with 6 sets of chromosomes. The genus Betula is a polyploid genus. They can have up to 10 sets of chromosomes in some of the species. And then morphological analysis of the dark bark birch population on our site showed a much greater affinity of sweet birch with of roundleaf birch with sweet birch than with yellow birch. And here we have the leaf silhouettes, Betula alleganiensis here, doubly serrate leaves, and then Betula lenta sweet birch here, single finely serrated leaves, and then here we have sweet birch. Roundleaf birch, excuse me. And then now below then are the bracts to the female catkins on the right, Betula alleganiensis, the yellow birch here, Betula lenta in the middle here shown, and then on the left is Betula uber. And even though on average Betula uber bracts are a little bit smaller than they are in lenta, if you look over the range, there's a lot of overlap. So statistically you can't distinguish between uber and lenta on the basis of reproductive characteristics, and most of the birches are differentiated on that basis. So you can't do that. It really is the leaf differences, but I put a note here too. We have not quantified the tree form branching habit. And that Betula uber, I mean, we look at these trees, you clearly see, I wish I'd had a better photo of it, that Betula uber has much finer branching than Betula lenta does, roundleaf birch, much finer branching. We hope to try to quantify this and hopefully using LiDAR technology to do that in the with leaf off in the future, another project that needs to be done. So morphological analysis of about 10,000 progeny of roundleaf birch and sweet birch from open pollinated mother trees. We conducted that in 1982, and over 99% of those progeny exhibited leaves typical of either Virginia roundleaf birch or sweet birch. Virginia mother trees, roundleaf mother trees average about 3% roundleaf progeny, and sweet birch about 0.3% roundleaf progeny. The frequency of roundleaf progeny among roundleaf mother trees, it was inversely related to the distance between pollen sources, as you would expect. The closer two roundleaf individuals were together, the higher the percentage of roundleaf progeny. This then led us to the hypothesis that roundleaf birch, that the roundleaf shape is a case of simple Mendelian inheritance resulting from the mutation of a single recessive gene with perhaps a few modifiers in the sweet birch population. And at the time, I found that to really be interesting in the light of a then recent article by Hunter Helu, who's with the Virginia Tech faculty on the role of single gene mutations in the evolution of flowering plants. It was really a great article summarizing the literature and demonstrating the importance of these single gene mutations in evolution of flowering plants. We subsequent control crosses between roundleaf birch and sweet birch in situ. We did those to try to refute this hypothesis. And so we produced over 8,000 progeny over 10 years, utilizing 2 roundleaf birches and 6 sweet birch parents. But we had, and this is another real problems. We're doing this in situ on these adult trees with ladders, with adverse weather conditions and then contamination from unknown sources. And so a lot of that data, most of that data was not usable. Exception was in 1986, although limited in scope with 2 roundleaf birches and 3 sweet birch parents. What we found here, here are the results here. We've got a U indicating roundleaf Betula uber, OP is open pollinated, and a lenta is sweet birch. If you look on the open pollinated uber, they're almost all ovate leaved, lenta-like leaves, right, with 2% intermediates. Down here at the bottom, an open pollinated lenta, which has 100% ovate leaves, lenta leaves. But then if you look at these 782 uber, when it's crossed with one of this tree 370, it's almost all roundleaf birches. There are 5% here, leaf shape of ovate leaf shape, lenta type, and 80.85 intermediate. So in contrast to then, these other 2 lentas, you get 100% lenta times this 100%, and then I'm sorry, this one uber 100% ovate leaves, indicating that it's a little a big A, right? That it has an uber gene in a cryptic state in it, and then but these 2 lentas you get a 50-50, indicating here that these lentas, you've got a gene there, right? I may say that wrongly, that this is 100% ovate leaf. So this individual's big A doesn't have an uber gene in it, whereas these 2 do. So we ended up concluding sample size is fairly small, but that it didn't refute our hypothesis that this thing seems to be controlled by a single gene. The results pointed then to the need for the establishment of a clonal seed orchard, and we did that at the Reynolds Homestead Research Center, and it at one time contained 119 individuals of roundleaf birch, sweet birch, yellow birch, and bog birch. We wanted to throw that in the mix. We did this to analyze progeny control crosses for for various morphological and chemical differences, DNA analysis to try to really identify that gene in Virginia around Lake Birch and their extent in the population. But as of May of 1994, there were 78 individuals that were still alive. But then the money dried up in 1994. No official records after that, and the orchard was later abandoned due to lack of funding for upkeep, and was really influenced by Peter's passing in 94 and not leading that effort. And just another one of these human factors involved in what happens in the recovery of species over a long period of time, or just the management of species, long-lived species over a period of time. Fertile grass on seedlings in 1983 failed from that. We were not successful with that on the seedlings. And therefore, clearly additional crosses need to be made to test our hypothesis. Retention of then this business of retention of existing germplasm through cultivation. I want to spend much time with this, but it began in 75 with the National Arboretum transplanting 3 of the seedlings there and producing vegetative cuttings for about 50 plants and distributing those to various arboretum, botanical gardens, et cetera. But we wanted a much more genetically diverse pool out there, so we mass propagated from seed in 1987 to Virginia at the Reynolds Research Center again, and produced over 2,000 seedlings in 1988 from control crosses that again were polluted, so we couldn't use that data really, but we distributed these very widely, including to the public and including to local citizens. And this was all under the regulations of BDACs, 3 seed plants per recipient, et cetera, that they went out. And so more than 800 individuals were distributed between 89 and 94 to more than 200 entities or location. And I said this earlier, no vandalism to additional populations following that distribution to local residents in 91. We also had outplanted 980 plants at the Reynolds Research Center in refugia planting, it was called, and 72 of those are still alive. So there are lots of individuals out there and about. And then finally, implementation of educational programs. I'll just simply say that we initiated, as I said earlier, and initially it was done to try to, first to really educate people about endangered species in general, endangered plant species. But then because of the vandalism, we began to back off on that, and we backed off because we thought it was bringing too much attention to the species and therefore could lead to vandalism problems. And it may not have reduced it. We can't prove it, but it stopped it, but it may have reduced it, of course. So next steps. Release of so these are things that we need to do, among others. Release of competition from the original population, and regeneration and treatments in that original population, because even though on that private land, and the private owned landowners now are very cooperative people now, that land ownership turned over, because we have that Lenta population in place there, and the Virginia roundleaf birch gene is most probably there in its cryptic state, most likely there, given the number of birches that are there, with some of those being heterozygous with respect to that gene. Continuing monitoring of additional progeny populations in terms of catkin production, diameters, and heights, crown class, and mortality. We need to quantify this branching pattern between the 2 species in the additional progeny populations. Regeneration treatments, we've only done it for 1. We need to do 19 more of these things. And then fencing and competition control in additional population 3 to enhance survival. We'll do that fencing this summer, this late summer probably, and that's going to be a real job. So if anybody's interested in joining us, that would be a good time to join us. Establishment of a seed orchard to conduct controlled crosses between roundleaf birch and sweet birch to further test our hypothesis. And then the question came up, is it possible then, as a part of this, to induce precocious flowering in seedlings to accelerating controlled crosses? I know that the American Chestnut Foundation research farm in Meadowview, which incidentally is only 30 miles from this I should have said this already, 30 miles from this location, are experimenting with this, and this would be something that would be very useful to accelerate this whole process. So by way of conclusions, to date, there have been some successes, and the first Betula uber is only one of two species that can be quote unquote considered as having been reclassified from endangered to threatened, primarily because of the ESA. And the argument was that other species had been declassified, but it wasn't mainly driven by funding from the through the Endangered Species Act. And secondly, here that Betula uber is considered, and I quote this, perhaps the most successful case of assisted colonization of a plant species under the ESA. So I'm trying to get a better handle on this up through the present to see whether or not these still hold, these statements hold. But at that time, that seemed to be the case. And continued, and I make the point here that continued success will depend on the degree of collaboration among various entities, and that ultimately probably funding levels to reach delisting, I estimated anywhere from $250,000 to $1 million, depending on the level of volunteerism and in-kind contributions. And I say including young people. I'm an octogenarian and therefore in the fifth and final quintile of life, as they say. And so we need younger people who will be around for another 50 years. And I make this point that long-lived plants require long-term plans, strategies, and actions to sustain them. And then finally, why should we care about Betula uber, other than the fact that it is treated under the law as threatened and endangered at the federal and state levels, respectively? Well, I turned to some of the words here of my colleague Heron Way, a forest geneticist and with a specialization in bioinformatics, and he offered up the following: genetic diversity. It may harbor unique genetic variations that contribute to resilience in a changing environment. Ecological stability as a distinct population, it could play a critical role in its habitat, influencing broader ecosystem dynamics. Evolutionary potential, some traits exhibit evolutionary reversibility, and protecting this population ensures that its genetic material remains available for future adaptation. And then finally, this thing of hidden genetic and molecular value, changes in leaf morphology, and I might add crown branching patterns, may indicate significant underlying molecular modifications affecting multiple signaling pathways, gene regulation, protein-protein interactions, and chemical interactions. And these changes could have biological significance that is not immediately apparent, but may hold long-term value. There's a lot of interest on the genetic control of leaf shape and various other characteristics currently. So that's it. Here are pertinent references that you can turn to. And then finally, here's my contact information. And again, please contact me in addition to capturing in the Q&A and what we talk about in these some minutes that we have left. Please communicate with me with your ideas and thoughts and willingness to get involved in some form or another. So with that, Sarah, I'll turn it back to you. And I guess do you want me to stop the sharing?
Sure, yeah, that'd be fine. Thanks so much, Terry. That was really fantastic. We've got some great questions, and I've got a few of your former students too. It looked like Susan Travis, and then Bernie Coyle, who is actually very involved with us here in TACF. They all wanted to give you a shout out. So
yeah, well, that's great. And Suzanne needs to tell me where she is because I haven't connected with her in years.
she said she's in Florida. So
Okay.
yeah.
She needs to
Hopefully.
send me her email address.
Yeah, I hope she will do that. I got a couple questions. One of the first ones that I think a lot of people are really interested in, I think you addressed this a little bit, and Vasily did too in some conversations and some of the advertising we did. But Helene asks, in case this isn't addressed, what are the lessons and insights that could aid work on the American chestnut?
I think a lot of planning and thinking upfront about how to do these reintroductions. there's a lot of ecology and a lot of genetics involved in that, and so what's going to go out there? Where is it going to be? How are you going to, how much effort are you going to be put into um, trying to maintain these populations by doing manipulations versus not doing manipulations? Clearly, if we had put those additional progeny populations out there and not done instituted some management practices, then there would have been a very different outcome. Now, if you have enough money, of course, you could do controlled experiments, right? And manipulate in some and not manipulate in others, and see what happens. And so I think I think that is a main lesson in all of this. And and American chestnut obviously occupied a very wide range of sites. So making sure, I think, in those initial distributions that you represent that wide range, that wide distribution of sites over which that species occurred. So yeah.
Yeah, good. And in my shout out from Bernie, Bob Ford, I see you on here, and I wanted to make sure to include you on that shout out too. So thank you for coming. And Terry, I don't know if you want to introduce anybody else. I know Emily and Ami were able to say a few words, and,
Yeah.
and you shouted out Hairong, but anybody else you want to?
Well, I did say somebody, but there I see Suresh up there, and he's been incredibly instrumental in terms of the GIS applications of this. He's a plant ecologist, and he's one of those younger people that is going to replace me in this equation. And he's already committed to that, and I'm holding, and now it's captured forever. And so if he abandons this effort, he's going to be in deep trouble. And Bob has been with me since the beginning here and did the dispersal work with Betula that allowed us to determine how far these seeds are distributed and in what directions and so forth. That informed us. And Vasily, of course, I mentioned too before in terms of his people helping with the catching counts. And I'm really hoping to really move forward with the foundation in terms of actually doing some stuff on the farm, perhaps putting the clonal orchard there, doing the control crosses. They're clearly much better at doing this control cross thing than we were in the past. And the business with precocious flowering and so forth, all of that's great. Kim Shear got some funding from the Forest Service, and Kim did the seed dispersal work so that we could see what the dispersal patterns look like relative to the patterns of seedling establishment. So all of those people, yeah, all of these people have been great contributors.
That's awesome. And Terry, Dana Nelson, who works with the Forest Service. Dana, thanks for posting and giving a shout out to the Forest Genetics 2025 Joint Conference, which is going to be here at Penn State. And he said that you guys should consider presenting that here. So please come to State College and present that work. I think, it would be really great to connect with some other colleagues. It's gonna, so for everybody else, that conference is open. Dana put a link in the Q&A, and I think maybe I can work with Hal to put that link in the advertisement or in the follow-up for this recording too.
When is it occurring?
It is occurring June 24th through the 27th.
Through the 27th, okay.
And I will be leading tours during that session, and also I'm on the career panel. So,
and this is what meeting again, is it? The what?
This is the joint meeting of the Western Forest Genetics Association, the Southern Forest Tree Improvement Cooperative, and then the Northern Forest Genetic Association.
Okay, yeah.
So it's all three forest genetics groups coming together for, I think, the first time. Dana is leading a 5K, which I will be participating in.
Great.
Well, so.
And so I'll be given 15 minutes to do this, and so I better get rid of a whole bunch of slides.
Right. Well, we are accepting abstracts and posters and everything right now. So head over to to that website, and I hope you all will participate.
Okay, great.
So let's see. William Truax asks, it seems that this tree exists at the forma or veritas level rather than at the true reproductively isolated species. Is that right?
Oh, absolutely. Yeah. That's absolutely right. I mean, clearly it's exchanging genes with quote unquote Betula, right? And And, you know, that's why it has varied so much in terms of how people have classified it, all the way from species right down to a form, which is I mean, and if I had to classify it, if I were in the business, and realistically, I would classify it as a form, as a local variant. I mean, subspecies are typically in trees or things of widely geographically distributed and overlapping just a slight amount with each other. Sometimes they're considered good species. And then moving down to from subspecies to variety, which are usually widespread variants of a given species, and then form is really a local variant. And that's where I would put it. But the issue is, you know, it's of course still officially recognized as a species, right, under the Endangered Species Act. And it's embedded in the Virginia Plant and Insect Endangered Species Act. And it's the only species that was listed as endangered under the original act. It's codified in that act. And it was along with ginseng, which was listed as threatened. So it's there. It's stuck in that act. And it would take an act of the legislature of Virginia to change that, to even to downlist it to threatened, which it hasn't been.
Yeah. Interesting. All the
Yeah,
implications there between the
yeah,
ecological and the legal.
yeah.
Um,
But
and
let's
you,
think about the importance of these single gene mutations, you know, and I think that's a really interesting conversation. And I know I thought Herong was going to be on here, but he's done a lot of work around that from a genetics level and, you know, the hierarchies of gene control and all of that business. And it's so he's very interested in carrying on with this.
yeah, that's great. Well, so kind of along those lines, Anita Klein asks, if the original population was very small, are you concerned about not having enough genetic variation needed for resilience?
Uh, yes. Yeah, but that's why we really, if we really could analyze these things, you know, vegetatively, and we're able to determine how widespread that gene for that roundleaf gene is in that population in a cryptic state, then we'd have a better feeling. How many of those trees that are out there, lentos, are really heterozygous for that? I kid my genetics friend and say, when are we going to be able to just walk up to these trees and put a probe on the side of the stem, and voila, there it is. The gene is right there. But yeah, no, it's a concern. But with, you know, we try to, but keep in mind that in those additional populations, those progeny from those 10 trees or 6 roundleaf birds originally were open pollinated progeny. So they were, you know, there's a lot of diversity in those 20 progeny populations, a lot of diversity.
So Kim Scherer pointed out that Hairong is in the audience. He's actually a panelist.
Oh, I see. I didn't switch over to do that.
Yeah, well, he also has a question. I don't know, Hairong, if you're able to ask your question. If you are, unmute and do it. If not, I'll do it for you. I'll give you a second. He might be doing something else, which is totally cool. He had a question about the catkins. Why were there no catkins in the spring of 2024?
We don't we think it was anecdotally at this point, think it was because of drought
Mm.
and because of a lot of other things were not in fact producing fruits. You know, all the hunters are out there and they're not seeing it. We have one local weather station and we're going to look at that. But here's another lesson for the future of American chestnut, and that would be to put a micrometeorological station out at these various locations. I would love to would be really, really highly valuable to have micrometeorological stations at each of these locations to see what's going on. And through time, of course, that's a thing that, boy, with mega dollars, it would be really worth doing. So, yeah, but
Nice.
probably drought, but unprecedented. We're going to go back and look at the data from that one station and see if in fact that year in terms of drought was unprecedented from the year before, because keep in mind that these primordia are differentiated in the year before, right, in 2022. And so they didn't develop. The males never fully developed.
Yeah. Well, I'm going to ask one more question. Before that, Terry, everyone, thank you guys for putting all of this together. Dana has put, so Forest Genetics Conference 2025.com. That's an easier URL to remember. Forest Genetics Conference 2025.com. It is open to the public. Please come. I think it's going to be a wonderful conference. The price is exceedingly reasonable. So I think you all should come and join us here
That's great.
in State
Great.
College at University Park. The
I
last
was
question.
around when that thing started, but go ahead.
Okay. Well, the last question, there was a lot of interest in vandalism. What caused it? Why? Can you talk a little bit more about that? And because I think
I
people.
think it started with, I mentioned with the National Arboretum taking 3 plants from the one private landowner. They said that they did it with his approval, but he later argued that they didn't. And then, of course, he's in the local community. He's talking to other people in the community. They're bad-mouthing this whole situation. And I think it just caused the whole thing to get out of hand. And it was out of spite, and he wouldn't allow us access to his individuals. He, in essence, by allowing us not to open up the canopy over his individuals, killed those individuals.
Mm.
I mean, but there was no convincing him. And so this thing of how the lack of protection of plants on private land, endangered, threatened species on private land is a real problem.
Well, and we've experienced vandalism too. Most of it's in New Jersey, which is no surprise, I think, to anybody. I'm being totally serious. Like, we have had trees cut down, we've had trees stolen. Like, it's definitely a concern.
Yeah.
Well, I'm afraid that that's all the time we have. Terry, there's a few more Q&As in here. I will send those to you. You'll
Great.
have everybody's email address to
Great.
answer them back if you're able to do that. Really appreciate everybody coming again. Next month, again, we're shifting it a week later. We're going to do March 28th, where Camilla and John are going to be here from the University of North Carolina in Asheville, and we'll be talking about entomology and chestnuts with a focus on chestnut weevil. So in the meantime, enjoy the rest of this winter and possibly early spring, according to the groundhog. And until next time, have a great one. Thanks, Terry.
Thanks to everybody.
Bye.
Bye.